Well, welcome to this little webinar. Under the fall conditions we had this year, uh, we have some questions about seed viability and vigor. And we have a product called Active Prime, which has a lot of good nutrients that would be useful for vigor. So we're just starting a project with 2020 Seed Labs to take a look at whether we can actually do anything to improve the germination and vigor of seeds, because of the poor conditions we basically had for both growing crops last year and then finishing them to get the seed really mature. And as a result, we're seeing some fairly low germinations and even lower vigor. Now, germination tests generally are done at about 20 degrees centigrade, so they're under good conditions. With the vigor test, you're putting that under cooler condition and stressing the plant, and we're seeing some fairly low vigor. We don't necessarily know what that means for sure, but we're gonna try and find out. And then the next question is, can we do anything about that? And we're wondering whether Active Prime, under the conditions that we've had, would really enhance those qualities of germination and vigor in the plant. And I want to just talk now about how a seed actually forms and how it germinates, and then we'll have another webinar or a follow-up once we get this research done and the results done, and that'll be give folks plenty of time to decide whether maybe an Active Prime this year is something you really want to do if you haven't in the past. So let's take a look at the presentation as I put it together. We know for a start that when we look across a field, there is tremendous variability. Now, this is a canola field, but we're going to talk primarily about wheat because we really don't treat a lot of that canola all the time because the industry kind of looks after that. But we do know on canola, for example, that the bottom parts generally have bigger seeds, and I believe that they may actually have the potential of having more nutrient volume in those seeds. Although the industry is basically looked at it and said, well, you know, small seed and big seed, really no difference in terms of quality of the seed, but now it's bagged so that you know exactly how many plants you're getting. We kind of don't do that with wheat, but we know that the variability across this, for example, up in the hilltops, moisture is different, soil quality is probably different than the foot of that particular slope. And we know that nutrients like sulfur can be tremendously variable, so can nitrogen, depending on, you know, denitrification and so on. So we know that the plant is dealing with variability in the field as well as variability along the spike when we're talking about wheat. So we get this mess of wheat growing there, and people don't take a lot of time to look at how an actual wheat head grows. The wheat seed starts long before you even think it starts. For example, here's a wheat head at stage 31. It's 1 centimeter above the soil, the first node. The seed head was formed way back even here at about the 4 or 5 leaf stage. Tell you the honest truth. At this point in time, this head already has all the seeds it can ever have, and now our job as farmers and agronomists is to save every seed possible. And here we can see stage 32. The node is now 2 centimeters above the soil surface, and the seed head just grows. It keeps growing until we get this head. Now, I want a little terminology on the spike, which is the overall head, call it a spike or a head. We have a group of seeds that is called a spikelet. That spikelet, and each one of these is a floret or becomes a seed. And that would be more important when I look inside the seed spike to look where this group of spikelets or the seeds are sitting. Here's a closer look at it. So we have one seed here, one seed here, one seed here, and this is a glume, and this is a glume, and then these are the awns and the lemmas and pellets. Now, if we look at the seed set, and this is on a tiller, you generally get 2 seeds, maybe 3 seeds, and they're kind of close to the same size. On a main spike, you're going to get more. You're going to get 3 to 4 and the odd time 5. So you can see in this one where we have 3 spikes, and you'll see when I pull a whole spike down and look at all the seeds, you'll see how this fits later. We have three. This one is bigger than this one because this one's pollinated first, this one's pollinated second, this one's pollinated third in that spikelet, and pollination starts in the middle of the head. Here's four. You can see these two seeds are about the same size all the time. This one's a little tiny bit smaller, and this one is the smallest. So what we have going on here is that because these four fit on this rachis node, these two seeds get nutrients first. And when a plant pollinates, it hormonally changes. And up until pollination takes place, the plant has a source, which is photosynthesis and sugars, and the sink is all the vegetative part and all the seeds and so on and anthers and pollen that are growing. But when it pollinates, hormonally the plant changes, and so does the sink. The sink now becomes the seed. So the plant then, knowing it's going to die in a short time after pollination under our climatic conditions, and that's based on hormones and photoperiod and temperatures and so on, the plant has a genetic program to die. It's a program called programmed cell death. So as a result, it's moving nutrients, and these two seeds get the nutrients first, and then these pull it later, and then this one is pollinated later, and it gets the tail end of it, and these tillers are even further down the line in terms of access. So when you look at those and pull them out, you can see exactly what's happening. Now, as the seed matures, you can see in this case, the germ is starting to form, and it's formed, and you can see that it's still green, and the endosperm is set up. Now, if there is frost or photosynthesis, long cloudy periods, stress on the plant, that's when we can have a reduction in the nutrients going into the seed, and we can have issues with quality of that seed if it's not fed properly during that period of completion all the way to hardening. So eventually the seeds get hard, the glooms and so on, and the tissue around the seeds, loosens so you can combine it, and we have our seed ready to harvest. Now here's where it's at, and I talked about this. It sits on this rachis node right there. All four of those florets are fed through that node. So the first two get the most of it, and then as the plant has been pulling all the nutrients and runs out of nutrients, if it's not really well loaded, those last seeds have less nutrient, in my opinion. And that's what we're going to kind of find out. But it's probably true, and it's variable. Now let's take a look at actually how the seeds set in the head. And this is a primary head, and this is a tiller. And what I did is I pulled them apart and set them out like this. And I did this years ago and used to talk about it, but gave up talking about it, went on to other things. Now take a look at this. So here's in the middle of the spike, 4 seeds, 4 seeds, 4 seeds, 3 seeds, 4 seeds, 4 seeds. As we move further down, you go to 3 and then 2s. And towards the top, we get 3s, 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 2, 2, 2. When you get drought, frequently you get all of these that will be aborted. The top will be white and they'll be all dead. Here's the tiller. 1s, 1s, and 2s and 2s. Generally, we sort of, if we could, we wouldn't want the tillers, the seeds, because they're probably smaller, and we'd want to kick out at least these last ones and this little one there. But we don't do that. So as a result, just by position, and whether it's a primary spike, whether it's the first or fourth one that is pollinated, or it's a tiller, we have differential nutrient density in the seed and probably quality. So here are the 4 seeds. If we could, we'd have to, if we wanted to kick out this one, we'd have to get rid of 1/4 of all the seed that we're producing. And if we got rid of all the tiller seeds, we'd have to get rid of a third of all the seed we harvested, and that's not what we do. So we know we got variance in the seed. So let's take a look now when this seed germinates, because this is where we're at now. What happens, the first thing that happens, the seed has to absorb water, and it has to absorb about 40% of its weight in water. In the seed, the only thing that is alive, because the endosperm is dead, starch, is this little thing called the aileron right here just under the seed coat, and the germ. The rest of this is just dormant sugar called in amyloplasts, which store the starch. And what happens when it imbibes water triggers the formation of gibberellins, which then trigger up the alluron to produce alpha amylase, and the proteases then produce beta amylase that starts to break down all this sugar and starch, forming glucose, which is then fed into the scutellum. And I'm going to show you all this stuff, but I'm using this diagram right now so you understand the process that will take place. The scutellum then feeds the nutrients from the endosperm into the germ, and we get germination. Before we get to show that, here's a slide that's used frequently, and Dr. Ishmael Chakmak developed this, and it shows zinc specifically. When we know work that Dave Hume did in Ontario as well when I was with Agritrend, showed that the zinc accumulates in the alluron and the embryo, because the zinc is really a key nutrient in terms of enzymes in the plant, and a lot of enzymes like alpha amylase and so on and other enzymes require a lot of zinc. Now if we take a look at alpha amylase itself, we have some chloride in it and we have calcium in it. So this just tells you that even all the enzymes that are required in the plant require nutrients that the plant is picking up and storing in that whole developmental period towards the end of its life cycle. Now here is the seed, and this is one that just started germinating. It's about 24 hours after we put water to it, and we can see here that the alluron is this little fine line right here. That's all that's alive, and the germs. You can see that this is the scutellum I mentioned. You can see that the starch is starting to break down, and the scutellum has actually moved some of that nutrient from here into the actual shoot that is developing in the embryo. And if you even look right here closely, you can see that the first true leaf is already developed. When the plant is finishing its life cycle in the field, it takes some energy and it kind of pre-germinates the seed, because this happens so fast when you add water to it. It's sort of primed itself already so that it germinates fairly quickly in the springtime. We'll take a little closer look, and you can see this is the alluron. This little red line just underneath the seed coat is all that's alive along with that germ. And here again you can see a close-up of that scutellum and the first true leaf here in terms of this is the coleoptile, and I'll show that coming up, and this becomes the radicle or the root. I cut that open, there's your first true leaf, happens early, and then this will move up and break through that eventually. So how did that happen? Well, the wheat seed forms a node right on the germ, and you can see that the coleoptile moves up from that, and the roots, the radicle and the seminal roots form there. And if you pull that off, you can see that this is the scutellum looking at it from inside the seed, and then this is the shoot, and these are the roots. And that's how the seed germinates and how it feeds that shoot and root, and then it splits off here, and eventually the shoot and root are connected at the crown node. So the next thing to look at is that gives you a clue and why we're looking at maybe how a primer, if it's absorbed into the plant with the imbibing water, might be able to help with the vigor going forward. If we look at the nutrients in the embryo, you can see that the embryo does not make up much of the dry matter, and it's not broken down in terms of what's in the alluron, but it's very small. So it's really important in the embryo as to what's there. 4% sulfur, a fair amount of P for energy and ATP, and really the start of building cells and to build the DNA in those cells from phosphorus. Potassium, magnesium, look at the zinc level in seed. 20% of all the zinc is in the seed germ, just like I showed in Chakmak's photos. Copper, There's quite a bit in the endosperm, 7% there. Manganese, a lot of manganese in there, and a fair amount of boron. And we know that we got boron deficiencies right across a large chunk of Western Canada. And we have those same nutrients also in the endosperm, which is then given off, or as at the endosperm breaks down, is utilized by the plant. Now, why would, you have to ask the question, why is it important for some of these things to be in the embryo? Well, you saw that first true leaf is already forming. Well, when the first true leaf emerges, or until the first true leaf emerges, the plant can only feed on what's in the germ, what little bit it can pick up in the roots, which aren't very active. Until the shoot emerges, there's no photosynthesis. And as a consequence, there's no energy being produced. It's all feeding off the seed and what the little bit of roots can put into the seed, but it's not really being able to create any sugar. But as soon as it does emerge from above the soil, it has to start photosynthesizing. So you need manganese there to help break down that water molecule in the first stage of photosynthesis in photosystem 2. So you want to make sure that the leaf is going to have enough manganese to do that, along with calcium and other things. And you're going to want a lot of zinc for all those enzymes, and you sure need that P to get it rolling. The potassium is used to expand cells along with water. Cells are born very small, so to speak, and the way a plant moves and grows its cells is to put hydraulic pressure into them with potassium and water. So there's a lot of reasons why the embryo stores all those nutrients. If we can supplement those, and we have a fair number of those in primers, and there are many primers on the market, maybe we can influence the quality of that seed, even if it isn't well established through the endosperm and the embryo in the seed. That's a guess. So let's finish this off. We get the radicle coming out. We have this coleorhiza here, which maybe we can get some nutrient in through there. And then we have some seminal roots, are the first true roots. There are 5 seminal roots in wheat. 2 come out, and then the remaining 3 come out later. And then the coleoptile moves up to the surface, and the first true leaf is inside of here. And the coleoptile, as it goes up, protects that first true leaf, and it opens up once it's above the ground. So if we have nutrient, and you can see this is an old shot from a dye that we're testing, you can see that dye is on the coating of the plant, and some of that, if it's soluble, will move into the plant, and it's basically on this tissue as well. So maybe we can get in. This is, again, the same shot. You can see the disco is on there. We now have 3 seminal roots. This is the radicle moving down, and we got the 2 seminal roots, and this is the coleoptile moving forward. And here's the coleoptile coming through the ground, and you can see it opens up and allows that first true root to emerge above the soil. And basically, the process is done. So with all that variability in the seed, the question becomes is, can we sort out which seed is better than other seed? Can we treat it with a primer or some nutrients that could be absorbed into the seed as it imbibes that water, if it's soluble in that solution? And will that influence the quality? Can we bring up a seed batch that at 70%, for example, German, maybe Vigor, to 85%? We're going to find out, and probably in 3 weeks or so, we'll be able to report on that, and I'll do another webinar to update you. Thanks a lot for listening.